Good evening, all. Today, I'm going to give a review on whole genome sequencing and social network analysis of a tuberculosis outbreak. This paper mainly focuses on genetics and about how a machine learning technique aided to identify the source of the outbreak. It involves more of genetics terminology, and I try to simplify as much as I can. So, coming back, this is related to a scenario which occurred in Canada when there's a sudden outbreak of Mycobacterium tuberculosis in 2006. As we can see in the diagram, there's a sudden increase in the cases starting from 2006 until 2007. Initially, two methods were employed to identify the cause of the outbreak, which we are going to discuss shortly. Later, the results of mycobacterial interspersed repetitive unit variable number tandem repeat genotyping suggested the outbreak was clonal and there were identical patterns in all the isolates. MIRO-VNTR is a powerful tool for studying the genetic relationships of Mycobacterium tuberculosis isolates. Next, we are going to discuss about the conventional methods used, which are traditional contact tracing and social network questionnaires. Traditional contact tracing was used early in the outbreak for purposes of secondary case finding and source case determination, but they couldn't extract much information and then they used social network questionnaires in the form of open interviews to expedite the process of finding the cause. This questionnaire was focused on drug and alcohol use, residential history, and travel history. However, there was no accurate positive sign in identifying the main root cause. As we can see the depictions in the diagrams, uh, the results obtained from traditional contact tracing are less evident when compared with the social network questionnaires. The orange circles indicate the the, the cases who are tested positive for tuberculosis. Blue circles indicates with the contacts, and the purple case indicates the main source case from which the outbreak has spread. And whereas in social network questionnaire, there are much more links founded in the form of interviews, and this white circles indicate the social congregations from which the outbreak has been spread. But still, as there is still not any concrete evidence, then in order to investigate further, they have employed machine learning technique. So then they performed whole genome sequencing by a technique called restriction fragment length polymorphism. This technique is nothing but it's used for the analysis of unique patterns in DNA fragments in order to genetically differentiate between organisms. This was performed on all 37 isolates who were tested positive, and they identified 204 single nucleotide polymorphisms which are unique to the 37 isolates. Both the RFLP and MIRO-VNTR typing of the outbreak yielded identical patterns suggesting a clonal outbreak. The review of RFLP data from the same geographic area in the years preceding the outbreak indicated that the same clone had been detected sporadically in the region since 1995. But still, as there was still no strong root cause, then the role of machine learning came into the picture. The SNPs were then concatenated and used to construct a distance matrix based on their similarity, which was then hierarchically clustered to generate a dendrogram, as you can see in the picture. This is the dendrogram which is formed based on the similarity measures of all the isolates. The results from the dendrogram showed that there were two distinct lineages of Mycobacterium tuberculosis, suggesting not one outbreak but two simultaneous outbreaks, which has different lineages, lineage A and lineage B. The presence of two lineages was observed regardless of the technique implemented, as they observed that the outbreak has two lineages. But when they found out that both lineages had been present in the region for at least 5 years before the outbreak, suggesting that suggesting that there may be another factor triggering the outbreak in 2006 rather than a genetic change in the organism. So now, after hierarchical clustering is applied to the SNPs, they came to a conclusion that it's the cause of social or environmental factor that triggered the outbreak and not a genetic change. So now let's see the results achieved. Epidemiologic follow-ups revealed that of the 41 patients identified with tuberculosis during the outbreak, 25 had a self-reported history of crack cocaine use, suggesting that the use of crack cocaine may have played a role in triggering and sustaining the outbreak. They also identified increased transient living arrangements and alcohol use during the same period. Moreover, surveys of crack cocaine users describe findings that are consistent with high rates of tuberculosis exposure. And several tuberculosis outbreaks have been reported in crack using communities, and later they have taken proper measures to limit the number of cases. I would like to conclude by stating that as there were limitations in traditional contact tracing to identify the cause of the outbreak, the integration of social network questionnaires and hierarchical clustering of genome sequencing enhanced the investigation process of M tuberculosis outbreak, and they got to a conclusion. This integration of machine learning techniques with traditional approaches would help scale up the efforts in identifying the cause of the outbreak with more accuracy and within less period of time. Finally, considering the cost effectiveness of genome sequencing in coming years, this integration of machine learning with genome sequencing would be widely accepted and commonly used for identifying the source of any outbreak that may occur in future. Thank you.